Poznan University of Technology.
Okay, recording is progress. It's past 5 p.m. Let's start our meeting. My name is Karol and I'm coordinator of the discipline of materials engineering at Poznan University of Technology. First of all, I would like to extend welcome to our guest, Professor Zygmunt Gryczynski from Department of Physics and Astronomy, Texas Christian University, Fort Worth, United States of America. Some words about professors. Professor Gryczynski is a specialist in the field such as single molecule spectroscopy, super resolution imaging, fluorescence application in biotechnology and life science, fluorescence spectra engineering. He is author more than 300 scientific papers, many scientific internships, and the manager of many scientific projects. His scientific papers are cited more than 13,000 times, a lot of citation. We are very pleased to have the opportunity to listen today this lecture. Today's lecture is organized as part of the STER program, guest lectures at doctoral schools at Poznan University of Technology. And today's lecture is titled Fluorescence Based Detection of Biomedical Diagnostics and Imaging. I think that all this from me. Professor, I give you the floor. Thank you very much.
Thank you very much. I appreciate the invitation, and thank you very much to you and Dr. Ala. And at this point, I would like to extend my all wishes and everything best from Texas. So I will be talking today about fluorescence-based detection and biomedical diagnostics, and eventually a potential application in biomedical imaging. So before I start, I want to give you some simple indication what eventually where I'm coming from, and I have problem with eventually doesn't want to move. Okay, I think I got it now. So I come from Texas Christian University, which is a small private university in Fort Worth, Texas. It's essentially 11,000 students, a kind of quite old university as count for the United States, opened in 1873. So it's over 100 years old. Small, but with very big endowment. And as in any university in United States, football is one of the most important part of the university. And TCU right now is one of the highest ranked team in United States. And now it's in top 4. So we have a big hope for big games coming soon. Fort Worth is known for a number of things, but it's kind of staying in the shadow of Dallas, so it's not so known around. But eventually it's the place where we have Lockheed Martin, where F-35 is assembled. And this is the assembly line for F-35, which is 2 kilometers long. Impressive view when you look on this. Another thing which is quite interesting is the Bureau of Engraving and Printing. And US dollars are printed in 2 places. One is in Philadelphia and one is in Fort Worth. Also quite interesting place to go and see, where you can see eventually how dollars and all the important papers are printed. In the same time, it's an old city, and it's a lot of what we call old money because it was essentially associated with oil. So at this point, it's a lot of interesting museums, one of the best museums in the States and probably around the world, very rich, having a lot of places. And this is an example of Kimball Museum, which essentially is a museum which owns Picasso, Van Gogh, all quite a few interesting places. And eventually, from the historical point of view, it's a quite interesting place because that's the place where Butch Cassidy and Sundance Kid were known to stay. And at this point, it's in the movies. You probably can remember this if you've seen it. And this is a present view of Texas, of Fort Worth, and at this point, it's quite sophisticated city. And this is Sundance Square. This is a center of the city where a lot of things happening, and it's a lot of cultural things going on right now. So now, eventually, what I am doing, what kind of technology, and what is my interest in the sense of my research. First of all, I grew up in Poland from fluorescence spectroscopy, University of Gdańsk. So fluorescence spectroscopy is my main interest. And my main focus is time-resolved fluorescence spectroscopy. And time is something very important with fluorescence. I will be emphasizing this quite a few times in today's lecture, because that's what we benefits a lot. And in this point, studying any kind of molecular systems, biological system in sense of fluorescence becomes very, very interesting for us. We also do a lot of practical applications in sense of multiphoton excitation or light quenching or still light stimulated emission, very important for super resolution. And besides this, we do some nanotechnology and engineering, and eventually searching for any kind of enhancement in fluorescence and enhancement of the detection and sensitivity, and in this case, in many cases, some specificity. And if you do fluorescence, you always end up with doing some microscopy. So eventually we did over time a lot of different technologies, starting from fluorescence lifetime imaging, FCS, single molecule, and a number of other things. But when you do the fluorescence and you want to do some stuff, the instrumentation is the most important part. And I want to show you a number of things which eventually we have and we can use and practically do the applications. Very important part, I have a good relation with number of companies, so I am kind of lucky to have a lot of different instruments and a lot of different lasers technologies which we can use for exciting the fluorescence and doing a number of different studies. One thing which I want to mention in sense of some kind of self-propagation, I want to mention the book we published with my brother in 2020. It's essentially practical fluorescence, and a lot of stuff which I will be talking about today, practical measurements, you can find in this book, and I will be eventually referring to this book in quite a few places. So it's essentially a hands-on experiments, and eventually books contains almost 140 experiments in fluorescence, starting with absorption and basic stuff, going to advanced fluorescence. So if you are interested in doing any kind of fluorescence experiments, you are very welcome to look, and you will probably find any kind of experiment you want to do. It's a lot of them. Now, talking about fluorescence, we need to understand a couple of things. One of them is a concept of light, because it's technology related directly to the light, and eventually identified any kind of processes which will be related to light-matter interaction. And in this case, what we have in mind, it would be if we want to have a fluorescence or any kind of response due to the light, we have to have a light absorption. So the system has to accept some energy from the light. And then we will have light emission, and light emission would be essentially divided to a number of different things, which we call in general luminescence. And I will come back and let you know what it really means, this term. And then part of luminescence would be fluorescence and phosphorescence, two different processes coming from different states. Fluorescence is a singlet state with transition, and phosphorescence is the transition from the triplet state. So let's start with light. And eventually for the physicist or engineers, the light is something which is electromagnetic radiation. What is important about electromagnetic radiation is essentially two practical vectors, which are electric and magnetic field orthogonal in phase traveling in one direction. This is extremely important because photon, as a part of particle of light, cannot exist as a standing photon, must travel. That's essentially the requirements coming from Maxwell equations. And at this point, if it's traveling in a well-defined direction later on, I will be talking about polarization, and this gives us a very important aspect of polarization. Fluorescence is essentially electromagnetic radiation. Hello? Fluorescence is electromagnetic radiation, which is a very narrow range, which is practically UV, visible, and a little bit near IR. But from the point of view of entire electromagnetic spectrum, it's a very narrow range. The range which corresponds to the energy, activation energy of most of the molecules in sense of singlet and triplet transitions. So what is luminescence? Luminescence, essentially a term coined first by Wiedemann in 1888 essentially comes from the Latin word lumen and describe any kind of phenomenon which is related with light. So emission of light from electronic excited state would be essentially any kind of luminescence. We have to differentiate this from a processes like random emission from type of Boltzmann emission, where you take a hot object will can appear red. It's a different type of emission. Here we're talking about the emission from electronic states. And we may have number of origin of luminescence, and at this point, this could be photoluminescence, radioluminescence, which is essentially an emission of light stimulated by high energy radiation like X-ray or alpha or beta gamma, electroluminescence, where its stimulation is an electric field or current, chemiluminescence, where chemical reaction gives us the energy to stimulate the first excited state, and then we can have the luminescence. Bioluminescence is essentially a chemiluminescence with biological reactions. Triboluminescence, which is related to the friction process, where eventually a physical force can stimulate some transitions and we may have some emission, or sonoluminescence, which is stimulated by ultrasound. But in case of fluorescence, it's part of photoluminescence, and it's essentially visible or invisible, which when it's in UV or near IR, emitted light by a molecule as a result of incident radiation of typically shorter wavelengths. I'm telling typically because with multi-photon processes, we may have a stimulation with longer wavelengths, but we have to use 2 or 3 or 4 photons. And at this point, the energy is still higher than emitted energy. So when we have an absorption of photon, the molecule goes to the higher excited state, typically at this point, a singlet excited state. And this absorption puts the molecule on the excited state. Single excited state is an allowed state. So in principle, in ideal conditions, the molecule can stay in the state forever. However, there is no such a thing like ideal conditions, because even in the gases, the molecule will interact randomly. But in typical solution, what we are doing in solution or any kind of solids where we have a strong interaction with the matrix or environment, eventually molecule will be deactivated fairly fast. And we may have a number of processes of deactivation where a simple deactivation can be by non-radiative processes, or in some cases, it could be a single transition from the singlet excited state to a ground state. And at this time, we have this phenomenon called fluorescence, because the access of the energy is emitted in the sense or in the form of a photon. And now we can eventually ask a very important question: how this process occurs. We have a molecule excited to a singlet state, and then it's coming back. I told you the process is fairly fast, but what is really important, the process is not instantaneous. Eventually, the molecule can stay for some time, and this becomes very important. It's a very short time, but this time gives us a lot of possibilities for practical application. So what is the fluorescence? Fluorescence is the light emitted by the atom or molecule after finite time duration subsequent to the absorption of electromagnetic energy. And this is the most important part, that we have this temporal break between absorption and emission. And this time is essentially let us learn a lot of any kind of system, what's happening with the system between absorption and emission of the photon. Another important part is the typically emitted light is of different energy, and it's typically a longer wavelength. But as I told you, in case of multi-photon processes, the wavelengths can be shorter, or upconversion technology, where it can also be shorter. Specifically, in most cases, practically 99.9%, and this is Kasha rule, which tells us that the emission is from the first excited singlet state. And this emission is a transition from its first excited single state to the ground state level. So after finite time of duration, the molecule gives eventually a photon, and this is a probability. Most of the molecules will go in non-radiative way, but in many cases, we have quite high, I will talk about quantum, yet we may have a very efficient process of transition to the ground state with the emission of the photon. But the lifetime, which is most important from my perspective, is the average time between absorption and emission. Since the time is dictated by interaction with the surrounding molecules, and eventually the same type of molecules in some cases, it's a random process regulated by the diffusion. So we have essentially a nice distribution over the time, and we can talk about average time, but we typically cannot talk about very specific time. So in case of fluorescence, for the allowed process, the emissive rates are very high, in the order of 10 to the 8, 10 to the 9. So a typical lifetimes of fluorescence would be in the range of 1 to 10 nanoseconds. It could be much shorter depending on the conditions, could be picoseconds, could be somehow longer when we have especially phosphorescence. But 1 nanosecond is extremely short time. It's a time where eventually light can travel only 30 centimeters. So it's a not big distance. It's something very, 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 very short. Fortunately, this time is corresponding to the practically any molecular motion of any kind of molecules, larger molecules, especially proteins. And that's what eventually adds to the value of fluorescence, because if these times are comparable, that's what I will show you, we can study and learn a lot about molecular systems. And eventually another process, which is molecule can go to a different state, which is quantum mechanically orthogonal state, triplet state. And at this point, the transition to the ground state is a forbidden transition, so the lifetimes are much longer, sometimes even in seconds. So eventually, I told you a lot about the fluorescence up to now, and I think you know much more than that. And question comes, why fluorescence? And from my perspective, from my old time, it's kind of simple, because from my perspective, it's a very pretty phenomenon. Essentially associated with light, we can see different colors, and it becomes quite beautiful. But from practical aspects, it provides a lot of information about molecular environment. That's the most important thing. And it provides information about dynamics of the molecular processes which are happening, provides information about polarities, pH, a lot of practical information, which we can learn just from studying the fluorescence. And at this point, when we have a prop or fluorophore in the environment, it becomes essentially a kind of a molecular stopwatch, which can give us the information about number of processes. Besides this, fluorescence can happen in IR, red and IR color, and at this point it becomes interesting for study or non-invasive studies of tissue. And this is the example of the finger of one of my students, which is illuminated with red light. And you can see this light kind of easily travels through the tissue. This gives us a kind of a way or gateway to our body without being essentially interfering with anything. And what is important, the red light is practically inert. If you go outside and on sunny day, you have plenty of red light, and we are inert to this. So using this kind of light could be really promising for non-invasive technologies. Besides this, fluorescence is very, very sensitive. It's easy to make a very low concentration. Picomolar, femtomolar are nothing unknown for fluorescence, and even single molecules can be done quite easily. The only requirements in case of single molecule is that the system must be extremely purified. But with today's detection technology, detecting single molecule becomes almost a routine. So let's come back and talk a little bit how fluorescence was developing. And I don't want to go through the entire history of fluorescence, but I want to give you eventually an idea how eventually a phenomenon evolved and where it started. One of the very first reports, it was quite a few reports before from Mandores and other guys in 16th, 17th century, but eventually a very important information comes from the Hirschfeld experiment, where eventually he reported that when he observes sulfide guanine, it's essentially a substance which was used for treating the malaria in 19th century. Eventually, what's happened, he realized, looking on the glass of solution of guanine sulfate, that it appears that when it's illuminated with the UV light, and if you're staying in a dark place like church where you have a lot of blue light, he observed it, and we kind of like to say through the glass of white wine, and white wine is a yellowish color. It works pretty nice like a yellow filter. He observes that the bottle gives some kind of celestial color. It was kind of interesting because he realized that the deep blue light can change the color upon interaction with the solution. He didn't know the explanation. He didn't know how to call it. And later on, Stokes took his experiment and said, that's what we have. And eventually we can observe this kind of emission and we can see a change of stage of the light. And that's what he didn't know how to call it. He at this time said, I will put the term which would be fluorescence. I don't like it because it's a kind of a strange word. That's how it was. I will probably find better one later on. And now 200 years later, we don't have better word and the fluorescence becomes a kind of common. So how it looks, this is a guanine sulfate, typical from this time from this time period. And when you dissolve this in the water, it's completely colorless solution. If you expose this to UV light, you can see this blue, nice celestial color. You can see this today if you take, let's say, if you go to the bars, sometimes in the bars, you can see this nice color when it's dark. And it's essentially tonic water, which the taste of tonic water comes from guanine sulfate. So if you take a tonic water, you put this on the UV illumination states, you will have this nice opalescent color. And eventually, after this, not much happens in fluorescence. And eventually, in 20th century, the big time happens for the fluorescence, essentially starting with Gaviola, where eventually he was the Argentinian scientist, which came to Berlin and did a lot of studies and eventually did first time resolved measurements. And at this time, he was doing this because at 1920s, epochal cell became valuable and he was able to modulate the light with higher enough frequencies, so he was able to measure fluorescence lifetime. Surprisingly, his measurements were pretty good. They are very nicely fitting to this, what we can measure right now. But that was essentially starting point where we realized that fluorescence is a finite phenomenon with a significant lifetime. Then in the same time, we have a quantum yield measurements, mostly done by Vavilov in Lomonosov. And he measured number of quantum yields at that time using his secretary, which was a young girl with good eyes, and she was able to practically count the photons. That's how precisely he was able to measure. And then later on, we have polarization of fluorescence, a very important phenomenon which gives a lot of power to fluorescence. Originally with the light polarization started with Wegener and eventually Perens, and huge contribution from Polish side where Alexander Jabłoński was the first one introducing the term of anisotropy. And I will show later on why this term is so important. And he did a lot of studies of anisotropy. And later on, in late 40s and 50s, fluorescence resonance energy transfer, another very important phenomenon, which originally was started by Perens. It's kind of interesting because both parents had the relation to Curie's, and younger parent was essentially growing up with daughter of Marie Curie, Skłodowska, in the same place in Sorbonne. And eventually big contribution from Forster, where eventually he calculated and he shows that this is essentially near field interaction phenomenon. And essentially from this time, not much was happening in fluorescence. We had some number of potential application, but it was interesting phenomenon only. But eventually in the 60s, a first work from Gregorio Weber essentially shows a practical application where he was able to develop dansylamide, which he was able to label the protein. And it's essentially in the same time, it becomes a very useful practical application. In the same time period, a little later, Stryer and Hogland were showing that Forster resonance energy transfer really depends on the 6th power, and Forster came, and Forster resonance energy transfer becomes extremely important. A few words how it's essentially how we look on this part on fluorescence, and this is something very important, which we call the Jabłoński diagram, where it tells us how eventually transitions happen. You have absorption of the light, which is extremely fast process. This is essentially 10 to the minus 15, 10 to the minus 14 seconds. It's this time because that's how long it takes for one wavelength to pass the molecule. And its perturbation is good enough to kick it to the excited state. Very fast relaxation to the first excited state. And from this first excited state, we have a transitions, non-radiative, which does not produce anything, just heat. And we may have radiative transition. And at this point, we have the emission of the photon. And we can probe with absorption any kind of excited state, vibrational state, or higher electronic states. And the emission always happens from the first excited state. And this is a principal Kasha rule, which tells us that emission will always happen from the same state, doesn't matter how we excite this. And this gives us essentially the idea of the absorption spectrum and emission spectrum, because the energy will be different, so we will have the distribution of the wavelengths. Another process, which I will not talk much today, is phosphorescence, where eventually it's an intersystem crossing to a different symmetry state. This transition is forbidden, has much longer fluorescence lifetimes. Now, some fundamental parameters for fluorescence, which we will be utilizing. So we have for fluorescence, we will have the emission spectrum, we will have the excitation spectrum, which probably should be first because this reflects the absorption spectrum of the molecule. This reflects where the molecule can be excited. And then we have fluorescence quantum yield. Surprisingly, it sounds quite easy. Measuring the efficiency of the process should be easy, but this is one of the most difficult parameters to be eventually measured very precisely. And then we have two very important parameters: fluorescence lifetime, which gives us very absolute information about the process, and it can be measured very precisely, and fluorescence polarization, which can immediately be used in practical application. And today, most of the practical applications comes from fluorescence lifetime and fluorescence polarization. How we can apply fluorescence for practical detection? You know, it's a technology which can detect a lot of stuff, but how we can detect anything of interest. So eventually, what we need to have the detection, or what is necessary to detect any object or process, we have to remember it could be also a process. In this case, in my case, I'm interested about any kind of physiological disorder, or simply talking about disease. What eventually we have to answer a couple of different levels. So how disease can manifest itself. In many cases, the disease can modify the local environment. So if we have a cancer, we know about hypoxia. Eventually, we know that this place is not so well oxygenated, so we may expect a different level of oxygen. At the same time, when we have hypoxia, we will typically have a little lower pH. So if we can detect this somehow, it could be a diagnostic element. In many cases, any kind of disease stimulates the release of so-called markers. Essentially, a dying tissue releases specific proteins to our circulation, and in this case, it could be any effect of misfunction or anything. And good examples could be cardiac markers like myoglobin or number of others, or cancer markers. Of course, they are in extremely low concentration because they are released to the circulation. And in case of heart attack, the heart is quite big, but compared to entire mass, is practically nothing. And if you have a small part of the heart dying, the amount of released proteins at this point is extremely low. Diluted to entire blood circulation becomes extremely difficult to detect. But that's what we have to look for if we want to do any kind of biomedical diagnostics or eventually imaging in the future. So what we will be looking for, if we have any kind of probe, if this probe gives us a signal, that's not a detection yet, because, you know, if you look on a white screen and you put a white paper on a white screen, it's very difficult to distinguish. So you have to have some contrasting elements. So the molecule has to respond with some kind of change when it gets to a specific conditions or when it interacts with a specific marker. And it could be anything. In case of optical technologies, it can be changing optical spectra, or emission spectra, absorption spectra, or change in the scattering. And for example, scattering can change when you have aggregation of the protein. So one of the way of looking on this is looking for the scattering. But it doesn't have to be only optical. It could be a magnetic or electromagnetic, like NMR, for example. It could be ionization radiation. That's what we use for the PET. Eventually, we have a specific delivery system which delivers the radioactive part, and we have essentially emission, positron emission out of this, and eventually we can observe this. Or we can have acoustic signal like ultrasonogram, where eventually we put acoustic wave inside the body and observe the scattering, and we can see different density of the tissue, and we can measure. And a number of others which happens. But in case of fluorescence technology, we will look for a number of things, and at this point we can hope for the change in intensity, where you have your probe interacting with any kind of market or environment. We can have a spectral shift or spectral modification. We can look for the fluorescence lifetime, which we can measure very precisely. We can look for the fluorescence polarization, or we can look for the change in the fluorescence resonance energy transfer. And this will be fluorescence resonance transfer would be really important when we have the change in the separation. But the changes in separation have to be very, very small, because it's a very precise technology, very limited to a specific distance. So what makes this technology useful? Let me start with something very simple. We can have some response to the environment, and this is what we call solvent sensitivity. A number of molecules could be solvent sensitive, especially sensitive to the solvent polarity. And here is a simple example where we can see the same compound in different solvent, like non-polar, completely non-polar solvent to a polar solvent, and we can see the color change, which immediately represents nothing else than change of the emission spectrum. What really happens? If we have molecule, which is essentially within the environment, and in case of fluorophores, we always have to think about molecule interacting with a lot of surrounding. So if you take a molecule of fluorophore in water, you will have plenty of molecules of water around. And if you come to the molecule which is in the ground state with well-established solvatic effect around, essentially the molecules are kind of balancing around, of everything moves. You have to remember everything moves in this kind of systems. And we come with the excitation, we excite the molecule, it goes to the excited state, changing in dipole moment, and now everything is rearranging around, and then emission comes from a little different state. And this difference essentially lowers the energy of the excited state, raises a little bit the energy of the ground state. So we immediately realize that the change could be quite significant in comparison with the light absorption and light emission. And this is a practical example of DOS. This is the molecule. It's typically stilbene-like molecules which can change the conformation. And we can see this in cyclohexane. It's very, very blue shifted, blue color. And as we change the polarity of the solvent going to butanol, we can find out it becomes practically red. It's a nice thing which can tell us about polarity, but eventually how we can build out of this a practical biomedical applications. And probably, I don't know how much you know about proteins, but typical protein will have a lot of amino acids, essentially putting everything together. And now protein is built on this way that it has a lot of polar parts, non-polar parts, and this is what eventual activity comes from. So eventually, if we take the example of a molecule like this one, and everything is moving pretty fast around, and you have molecule which sticks to this, if you take a molecule which is sensitive to the polarity, if it sticks to the non-polar side of the protein, which could be a nice example, it will immediately respond with the change in the fluorescence signal. And this is a practical example of one of the marker INS, where eventually, if you take the solution of INS, it's practically colorless, a little yellowish. If you take protein, this is HSA, you can see this blue color, but this is due to the scattering on the protein molecules. If you mix them together, it becomes nicely fluorescent system. So if molecule interact with the protein, this INS, you will find out the change in fluorescence. That's what could be a measurable effect. That's how you can detect the protein. And this is the example where eventually we put a different concentration of the protein with the same concentration of INS, and you can see how this changes with the protein concentration. And this was essentially a detection of binding, and we can see the binding of the INS to the protein. But in practical case, that's how we can detect protein. And one of the example could be Lugal, which is essentially used in CSI scene when they walk to the scene and they want to look for the protein or blood stains eventually, which are washed but and not visible. If you put this dye and it sticks to the protein, and it's not only luminol which does it, but it's number of other dyes like indocyanogreen or anything emitting in the red, and you put a UV illumination, you can see immediately high fluorescence, and you can localize any protein on a surface or any place. And this is essentially a description how these measurements can be done. And another example could be a protein, which is a nice example which I took from Joe Lakovich's book, where eventually you can take a tryptophan emissions spectral dependence on environment. And if you take a protein where tryptophan is very well shielded, no access of water, it would be essentially completely hydrophobic environment, and the emission would be blue shifted. Like we take azurin protein, which eventually gives us this kind of a spectrum. And as we move to a different protein, or protein unfolds, we will see the change of the spectrum. So the tryptophan, which is buried inside the protein, you will have a very blue shifted emission, and as it goes outside, you will see less and less, and eventually outside it's red shifted with much broader spectrum because of the broad range of interactions with water molecules. Molecular system sensitivity to local viscosity. That's another very important parameter, and I will be talking about this a little bit and give you some examples, because eventually what we realize, the viscosity is something which is quite important for any biological system. But it's not a classic viscosity which we understand by macro viscosity, where we can take a viscometer and measure the viscosity. The question is, what is the distribution of viscosity in any kind of tissue, or especially cell or even nucleus? And at this point, it's a different question. We cannot take this, we cannot measure this. So we are looking for the molecules which can report on viscosity. And one of the examples could be work which I'm doing with my colleague Sergei Dzuba from chemistry here in TCU. He is working with a number of molecules which can change the conformation in the excited state, so-called molecular rotors, which they can be planarized. When they go excite, they twist, and they change the spectrum or change the fluorescence lifetime. So this is one of the examples of protein rotor or body P rotor, which can change the conformation. And it's a number of works, and porphyrin rotor is pretty nice because you have a spectral change, so you can easily build what we call a ratiometric sensor at 2 different wavelengths, and you can detect the viscosity. We did a lot of different works. I listed here a few earlier work we did, but now we have many more. Now, in this case, you can see the change in the intensity at a given wavelength, and we can eventually find out how it depends on the viscosity. We can calibrate this, and typically this kind of calibration is done with the solvents which we know viscosity, and typically it's ethanol glycerol mixture or methanol glycerol mixture, where we can macroscopically measure the viscosity. We know that the system is continuous, so we don't have any local effects. And at this point, we can calibrate the rotor, and now we put this inside the cell or tissue, and we can measure the viscosity essentially on a molecular level with the resolution of optical microscopy if we need this. And in this case, we can have also a lifetime change in case of rotor, which is much more useful because eventually lifetime measurements is completely independent, and measuring the intensity or even spectral change can be perturbed by scattering a number of parameters. In case of lifetime measurements, you say, in some sense, ratiometric measurements, where eventually gives us an absolute lifetime, which does not depend on signal attenuation or number of processes, which could be not affecting the fluorescence lifetime, because if we have something like a dynamic quenching, this will change the fluorescence lifetime. And rotor works on this basis, essentially changing the conformation, quenches the fluorescence lifetime, and it becomes shorter. And lifetime we can measure very precisely, so fluorescence lifetime microscopy becomes very powerful. I want to show you one practical application in this case, how eventually it's happening. And for a long time, I was working with my colleague from University of Montreal, Richard Gryczynski. He works with mostly with cystic fibrosis or chronic obstructive pulmonary disease or asthma. One thing which you have to realize, most of the organs which we have in our body, and especially any opening in your mouth, is essentially covered with very thin layer of mucus. And you know, our cells produce mucin, which are special proteins, release them, and eventually this kind of layer of very viscous material, essentially a very thin layer of mucus, it's a protective mechanism. It typically protects us from debris or viruses or bacteria. Essentially, when we breathe the air with everything, a lot of viruses. And typically, all these bigger particles are immediately captured by this type of very viscous protein. And in this case, you probably know this from a bedside, because if you get sick, if you get essentially a flu, you realize that your nose can run like crazy. That's essentially a release of mucus from epithelial cells on your duct and on your nose. And at this point, they produce a lot of this mucus, and this is very simple reason, because this is the only effect in biology where granules containing mucins release this and changes the volume over 1,000-fold, or close to 1,000-fold. So eventually, any kind of tissue is built from epithelial cells. On top of them, it's a thin layer of mucus. And eventually, you have what we call cilia, which beats and eventually produce a motion of this viscous fluid up, which eventually comes out down to your nose. And that's how most of the viruses and debris are eliminated. So eventually, what's happening when we produce the mucus, mucin, we have a granule, which a special organella where mucus is built, mucin protein is packed, and when this granules comes to the membrane, it essentially fuses, releases, and eventually expands and forms the monolayer across. And now a question comes about the volume expansion and eventually viscoelastic properties of this kind of mucus layer, which is extremely important, for example, in cystic fibrosis. And we did a lot of studies where we were studying the mucus formation, and we can see these granules by loading this with acridine orange. And when we have high concentration of acridine orange, and this acridine orange happens, it was a coincidental finding that it accumulates in these granules, and it accumulates in so high concentrations that start aggregates. And aggregates of acridine orange, it's essentially a dye, one of the very first dyes. But if it forms acridines, it becomes red shifted, and it has very long fluorescence lifetime. It's a very unusual thing because typically aggregates have much shorter lifetime. But it was quite nice, and we were able to localize the granules and see how they there. And I don't want to show most slides, but eventually when this granules comes to the membrane, fuses, releases, diluting the acridine orange, eventually this red color disappears, and we can see how fast this eventually spreads on the surface of epithelial cells. But one more interesting studies which we did quite recently, let's say not very recently, but which we did, it was cystic fibrosis. And with cystic fibrosis, we have a kind of a long-standing question, because gene for cystic fibrosis was discovered over 25 years ago. At that time, the thought was eventually within a few years, we will have a cure for cystic fibrosis. Unfortunately, 25 years later, we don't have it. And the question comes, it's something happening which is very difficult to understand how it's really happening. We know that cystic fibrosis, the viscosity of the mucus which is formed, it's too high, it cannot be transported by the cilia, and that eventually becomes a problematic, because at this point, if it's not transported, it stays in your lung, produces water, and eventually that's what is the problem. So we did an interesting study with University of Maryland, where my colleague was able to collect the regular cells and cystic fibrosis cells. And we ask ourselves how eventually packing in the granules happening in these cells. And the expectation was, like from the old times, that when you have a cystic fibrosis, the packing of the mucin protein is probably too big, and the viscosity of the mucin protein is much higher. That's what we were looking for. So when we took eventually image the cells of non-cystic fibrosis cells, we find out that the mucus have a specific viscosity, which we can refer through the fluorescence lifetime of a rotor. That was a body P rotor. And at this point, we can find out how well they are distributed, and it's a pretty nice distribution. It looks simple, but it was thousands of cells analyzed, a lot of work to do it. And then we take cystic fibrosis cells, and we find out, which was a first surprise for us, we find out that overall viscosity is much lower. But we were able to change this distribution and analyze this in the form of two distributions. One, which is practically the same distribution of normal cells, and then we have much lower viscosity in this part. And that was a surprise. But eventually later on, we found out that it could be explanation for this. Probably when the mucin protein is formed in the cystic fibrosis cell in granules, the packing is not perfect. And because of some change in the mucin, the packing leaves some pockets of water in between. So this lower viscosity eventually reflects this open pockets of water where the rotor has much lower viscosity and can change the conformation much faster. That's why we see the fluorescence lifetime, which is much shorter. One more example of molecules which interact with conformational change, it could be DNA markers, or so-called DNA intercalator. And one of the oldest one is ethylium bromide or ethylium bromide dimer or TOTO or yo-yo. These molecules, when they go inside the DNA helix and binds to the groove or any part, are essentially immobilized and stabilized, and heavy quenching is essentially stopped. So molecule in the solvent is non-fluorescent. When it comes to DNA, it becomes much more fluorescent. And we can observe the change in the intensity and in the same time change in the fluorescence lifetime. And later on, I will tell you much more about practical application on this for the DNA detection. But this kind of a dice at the first plays a fundamental role when we are doing a genetic testing and eventually DNA, after DNA amplification, detection of the DNA, and practically DNA sequencing. That's how eventually it's done with this dice, and that's how the genome was done. Now, for a moment, let me come back to the light, and especially light polarization. A very interesting concept. I think it's quite powerful concept. If you look on the electromagnetic light, it always travels in one direction, and it's always electric and magnetic vector orthogonal to the direction of propagation. So very first interesting concept which comes out of this, we may have isotropic light, or something which we call isotropic light, but it's not really isotropic, because if you look on this distribution, it's isotropic when you watch this on the direction of propagation. But if you look on this on the 90-degree angle, you will find out that this is a highly ordered system. The oscillation is only this way, so it's highly polarized. We can have polarized light, which eliminates all the side components, and this is much easier to deal. That's why we can use the polarization, and we can have a lot of applications of light polarization. Just a simple case, you can have a polarizer. These days is quite easy because you can get very cheap sheet polarizers, and you can find out that they will polarize light with extremely good efficiency. You rotate the polarizer, you will find out completely different polarization. Interesting thing, we have something which we call Molus law. Eventually, intensity of transmitted light is proportional to the cosine square, and this is because of the amplitude of light and intensity proportional to the square of amplitude. But very interesting phenomenon here, if you cross the polarizer, so one is vertical, one is horizontal, you will find out that the transmitted intensity is 0. And it could be a surprise, because if between these two polarizers, crossed polarizers, what we call, you put third polarizer under some angle, you suddenly start transmitting a light. In case of perfect polarizer, if you put infinite number of polarizer, turn it by small angle, you will find out that in theory, we can turn the light polarization from vertical to horizontal, and that becomes a very interesting phenomenon. But what I want to point out at this point, we can polarize the light. Why this could be important? It could be important because eventually light absorption is associated with so-called transition moments. So if we take the simple molecule like this naphthol, we will find out that any molecule, we have high occupancy molecular orbital and low unoccupied molecular orbital, and transition between them is always associated with the dipole change, and this generates an electric dipole transition moment. So what this practically means, any kind of molecule will have some transition moment associated with this. If you take elongated molecule like DPH, DPO, number of them, it would be always along the structure of the molecule. If you take high symmetry molecule like anthracene, which is C2V symmetry, or any molecule which is C2V symmetry or D2H symmetry, you will find out that transition moments can be only oriented along the main symmetry axis. So they will be always orthogonal. And one transition would be only one dipole direction. So it will absorb only the light polarization along this direction, or component of light polarization about this direction. Any non-symmetrical molecule like indole or tryptophan will have arbitrary distribution of transition moments, but any transition to a singlet state, S1, S2, or S3, will be associated with well-defined transition moment. So what this means now in practice, if we take molecules which are randomly distributed, and we put an excitation, polarized excitation, we will excite the molecules mostly which are oriented parallel to the direction of the light polarization, and much less the molecules which are not parallel or orthogonal. They will not be excited at all. Eventually, as a concept of this, we are producing something which we call photoselection. So eventually we produce the most transition moments excited along the polarization of the light and practically none orthogonal to the polarization of the light. So eventually with photo selection, we orient this system, but we orient the system of excited molecules only. So we don't physically orient the system, but we select the molecules with the specific orientation. And at this point, it gives us extremely big power. Excuse me?
Можно на крышу?
Это ж я понимала, что это не бомба, но это...
If we have question, I can answer at this point, but
It's a problem. I please participate. Microphones, yes.
okay. So let me tell you now what it means, how well this polarization can look. Hello? At this point, it's a nice experiment, and now if you look on this picture very close, this is essentially observing the different molecule, let's say the same molecule under different conditions where you can have fast molecular rotation or slow molecular rotation, which does not depolarize. And in case of rhodamine, when you look through when you excite this in water, you will find out that if you look with the vertically and horizontally polarized oriented polarizer, you will see that intensity is identical. And in case of high polarization, the same molecule in glycerol, you can see definitely different intensities. And in the same time, if you rotate the excitation to the horizontal excitation, where you have a random distribution, they are exactly the same intensity. What this means, in practical application, if you do the polarized excitation, you photo select the system, and now you can measure two intensity components, vertical and horizontal. And eventually you can define the term of polarization. This was the first term defined after Weigert, where eventually we were doing the difference. That was for the light polarization. And I think it was in 30s when Jabłoński for the first time defined this term, which essentially relates the change in the intensity between vertical and horizontal component to the total intensity observed from the light. And this gives us essentially a measure of anisotropy. And what is really important in this case, you will find out that this can be related by essentially expressed by the ratio of these two intensities. And now if we can relate this to the ratio of two intensities, we realize that this parameter becomes a ratiometric parameter. And that's where the power of anisotropy comes. It's a ratiometric parameter, does not depend on any attenuation change in the excitation, light intensity, or anything which we can have. It gives us always good answer. And this becomes practically a very powerful parameter, especially when we recognize a parent law of rotational depolarization. And we find out that the lifetime of the molecule is comparable to the correlation time of the same molecule. Essentially, the parent law gives us the relation between the lifetime of the molecule and time correlation, rotational correlation time. And in case of this rotational correlation time in the parent equation, which is essentially later on defined by this Einstein-Smoluchowski equation with the viscosity, volume, and temperature factor, this is essentially a time which takes for the molecule to rotate 1 radian. And at this point, we can find out that the time of rotation of the molecule, when it's comparable to the fluorescence lifetime, the measured ratio of anisotropy or measured anisotropy may change significantly. What this means, if we take two molecules, one rotates faster, one rotates slower, when they have similar lifetime, they will give us completely different response in sense of anisotropy. So in some sense, we can exactly measure, if we know the lifetime and we can measure the lifetime, we can measure how fast the molecule rotates. Now, why this is interesting? Let's say first what's happening, if we come with the excitation, which is polarized excitation, we photo select the molecules, and now the molecules are photo select, and if they move, they essentially randomize, and eventually they, from photo-selected distributions, they become isotropic distribution. And what we observe the anisotropy decays, anisotropy changes in time. The very simple way of changing this is changing the temperature of the solvent, and we can see the dependence of the temperature. You can see clear dependence of anisotropy on temperature. But practical application comes in many other aspects. One of these, it could be eventually a lipid, a membrane, which is classic lipid. If we put a fluorophore molecule inside the lipid, it's typically well-oriented. The mobility is very much limited. And at this point, we will measure very high anisotropy. As soon as the lipid is unsaturated lipid, or we go to the transition, we'll heat up the lipid and eventually it melts, we find out that it becomes mobile and our anisotropy goes down. So we can eventually see processes in the membrane, how they happen. Besides this, we have a number of dyes which will respond to the environment of this kind of lipid, like proton, and a number of things which can be easily studied the membrane formation. Another aspect could be if we have a protein which is labeled or eventually contains, for example, tryptophan, which is a natural label which we can have. But if it's labeled, if the protein aggregates, the size changes, and at this point, the mobility of the molecule can change. So if we take a big protein like this one, or we take small fluorophore like this one, the protein rotates very slowly. It's essentially a tumbling, which is a slow process, and rotation of the fluorophore is pretty fast. But if they bind together, your fluorophore will only rotate with the protein with some local mobility, which could be allowed. And in the same case, if we have singular proteins which can aggregate and we form the aggregates rotates much slower. So we can follow these processes. So we can see the interaction of the protein as it happens in natural system. And this is the example where we can have essentially a association of tetramers, where we have a single monomer which associate to the tetramer, and we can find out that the rotation time changes dramatically from 5 nanoseconds for the monomer to 20 nanoseconds for the tetramer. This is a frequency domain measurement. That's how originally this was measured, but time domain is much simpler interpretation. So immediately comes to a very simple example where we can have anisotropy of a mixture. If you have a fluorophore interacting with the protein and you want to see the equilibrium between bound and unbound, you can follow the polarization of so-called anisotropy. The advantage of anisotropy is that anisotropy is an additive quantity. It's not only ratiometric, but anisotropy is essentially a sum of fractions and anisotropy for a given fraction. That makes it really, really simple. It's the same with the polarization, but the addition is very complicated. Here is much simpler, straightforward. This is the example how eventually this can be done. Use this mixture of the same of different fluorophores in the same solvent, where eventually they have different polarization. When you make the mixture, they are classic mixture, and then you can see how anisotropy changes from one to another. And now let me move to another phenomenon which is quite important in fluorescence, which we exploit quite a lot in any kind of biomedical application, which is FRET, well, for fluorescence resonance energy transfer, or for last 10 years, we call it Foster resonance energy transfer, to just give the credit to Theodore Foster, which was eventually for the first time describing the process in fluorescence, and eventually changed the applications of fluorescence in many aspects. And at this point, what it really means, if we have two molecules and one molecule has the emission absorption in more blue range and emits in this range, and you will have another molecule which will have absorption in the range where molecules emit, we will have so-called overlap integral, and we may have eventually energy transfer from one molecule to another molecule. What is very important, this emission energy transfer is essentially without the appearance of the photon, and it happens through the space. So if it happens through the space, will depend on the refractive index and number of other quantum mechanical parameters, but it will be very well defined for a given molecule. And what Foster really showed at this point, that the rate of energy transfer is proportional to the parameter which he calls R0, which is a characteristic distance calculated from this overlap integral. And it's typically in the order of few nanometers, definitely below 10 nanometers. We have very few examples for very long-lived lanthanides probe, which could be close to 10 or little more than 10 nanometers, but typically is below 10 nanometers. And it's depending on the ratio to the 6th power. So it's eventually decays very, very quickly. What it tells us, anything which is within the Foster distance, we will have a significant transfer. When anything which is off the transfer, Foster resonance of the R0, the transfer would be much, much less. This near field interactions is very, very powerful. And what is happening, most of the protein sizes, when molecules interact, they come in the proximity below 10 nanometers. So we can study a lot of proteins. And this is the example of eventually a simple thing. You label the protein with two molecules. One is donor, so-called donor, one is acceptor. And at this point, if molecules change the conformation, a small change in the distance, because first resonance energy transfer is extremely sensitive to small changes, we will see the change in the thread. And seeing the change in the resonance energy transfer tells us very, very precisely how the molecule changed. The first thing was essentially for the first time shown experimentally by Stryer, Lubert Stryer and Hogland. Hogland is essentially a guy which later on opened a molecular probe, essentially a technology which was essentially, he opened this here in Texas, in Austin. And his first molecule he produced was Texas Red, very famous, still used. And Lubert Stryer is essentially a guy which showed that energy transfer can be used in many aspects. And he kind of revitalized the first resonance energy transfer and essentially introduced in a broad range to biology and any kind of biomedical applications. And I want to mention this because Stryer is another kind of link to Poland. And eventually his family comes from the place in Poland, eastern part of Poland before Second World War, which was Stryj. And when they were escaping from this place during the Second World War through Shanghai, and he came to America with his parents decided we have to have some names. They had Jewish name. So they find out we are from Stryj, so let's call us Stryer. That's how it's happened. So it's another Polish connection in case of a first resonance energy transfer. So it's a lot of practical application. I want to show one very simple application where we're looking for the activity of MMP proteins. These are enzyme, cleaving enzyme, which are very active in case of cancer. If you have a cancer, eventually this enzyme shows up in the tissue and cleaves the proteins on a specific sequence. So we built a very simple sensor where eventually has a donor acceptor system linked by the specific peptide, which is sensitive to MMP9, it's an entire family of metalloproteinases which do the cleavage. And at this point, if it cuts, eventually it's a complete separation. Thread disappears immediately. And some spectroscopic results which shows how it changes. You have a quench system, and eventually when you cleave it, the fluorescence of donor is released, fluorescence of acceptor decreases. And this is a practical picture. And this is an uncleaved system, which is red color a little bit. And when it's cleaved by MMP9, it becomes green. We change nothing, it just drop of enzyme, cleavage, and you can see the change in color very, very easily. What makes practical detection difficult? I now move to a second part where I will show you a little more practical applications. Now, as I told you, the technology is extremely sensitive. Up to now, it's probably, let's say not probably, it's for sure most sensitive technology we know. But with the sensitivity comes also a problem, because if technology is sensitive, it detects not only this what we want it to detect, but detects anything which shows up. So we can do the single molecule detection, yes, very precisely, but it's only in extremely purified system in confocal volume. And confocal volume comes from this, that is only purified, but we have to limit the number of molecules in the volume which we detect, molecules of water in case of biological system. Because if you have a single molecule of fluorophore in a femtoliter volume, or even much less than this, we have still dominant amount of molecules of water. And Raman scattering out of the water may dominate easily fluorescent signal. So if you don't do the confocal volume, you will not see single molecule, because the noise scattering, Raman scattering, which is typically in the range of emission of this high quantum field red emitter, it becomes really, really problematic. But in case of biological system, we have number of aspects. First of all, if we have any biological system like blood, plasma, or anything, even saliva, it's a lot of different things. So we have to deal with a lot of garbage, and we cannot purify this, because as soon as we purify it, we don't need to do the fluorescence because we already separated the parts. And in most cases, purification is impossible. We want to do this directly in the tissue, and when we do this in the tissue, we deal with a number of different problems. We come with the excitation, and if it comes to the skin, it can penetrate. Let light will penetrate, or blue light even a little bit. And when you get the fluorescence, which comes back and it's scattered, all things in the middle will give you the signal. You will have the fluorescence. And at this point, we have to deal with something like autofluorescence, like leak of excitation. And you have to remember all the system, even here, if you take a plasma or anything, it's a highly scattered system. Blood, extremely scattered, tissue, completely scattered. Raman scattering, it would be a problem because any biology will happen in water. And on top of this, we have instrumental noise, which we can try to limit as much as we can, but it becomes very, very difficult. So what limits our application is essentially a signal to background ratio. And at this point, how we can improve it? We know this for last 50 years, that this is a problem. And for last 50 years, we try to produce brighter probes. And yes, we produce probably the most powerful probes we can have. Eventually, all the recent red dyes reach the limits of quantum mechanics. We have the extinction coefficient of close to 200,000, quantum yield of close to 1. But with this comes another problem. High quantum yield, eventually a red shifted dye, which is good for tissue. It immediately tells us that stock shift would be much, much lower. It's extremely low. And we come to the problem of scattering and all these other problems which comes with this. So we cannot improve anymore on this side. Then the only thing which left for last quite a few years is trying to limit the background. How we can limit the background without limiting our fluorescence probe? We have to ask ourselves question what eventually the background is, and can we do something about background? Most of the background components would be essentially high excitation light scattering, Raman, autofluorescence. And we have to remember, biology is dirty. Anything in biology, if you take tissue, it's everything. You eat something like in St. Patrick's here in the United States, we eat a lot of green. Immediately you have a red dye in your tissue, and you can detect this quite easily. So autofluorescence, and we cannot get rid of any kind of byproducts, which are proteins, flavins, porphyrins, heme is degrading and produces a lot of other things. So that eventually comes to everything what we have. So one thing which we have to realize, we try to take an advantage of longer fluorescence lifetime. What this really means, eventually it means a number of aspects where we can realize, and let me come back, most of this scattering is instantaneous, Raman scattering is instantaneous. Luckily, most of this autofluorescence byproducts, biological byproducts, are short-lived. So at this point, we can have some advantage. What we have, we can try to do long lifetime dyes. Eventually, we produce the dye which will have a longer lifetime, will give us huge benefit. What is good? We can easily use a gated detection. They will have typically large stock shift. So very nice. The problem is, in this case of long-lived dyes, that they have typically low brightness because the transition cannot be so easily allowed, because otherwise the lifetime would be short. And this can be done by lower extinction coefficient and lower quantum yield. We typically want to have better quantum yield, so we sacrifice the extinction coefficient. And at this point, what we can do, we can say we need just a higher concentration and we will be good. But if we want to study biological system, you have to remember you cannot load as much dye as you want, because it's typically toxic, and FDA and all these things will never approve it. And besides this, it's eventually modified physiological function. So you want to limit eventually the amount. But what you want to build, you want to have a dye which will have a longer lifetime, so you can use the timing to eliminate the short-lived background in sense of scattering of fluorescence. How it can be done? That's an example where you take essentially a fluorophore which has a longer lifetime of 20 nanoseconds and another which has 2 or 3 nanoseconds. And if you excite this, you have a signal 50-50, which is, from the experimental point of view, garbage. But if you gate this 12 nanoseconds, the short-lived component almost completely decays. You decay also, that's what is important. Gating is a passive technology, which also lowers the signal of your probe. But at this point, you live up with mostly with your probe. How it works in practice, it works. Let me change the pointer here. So in practice, what's happening, you have 3 dots here, which eventually have short, longer, and longer lifetime. And if I start, you can see when I shift the gate, you see that the fluorescence disappears quite significant. Here's another example where eventually these are short-lived on this side and it disappears very quickly. And here is interesting example which led us to a number of applications. We put essentially a tubing with long-lived dye, which is in this case ruthenium complex, which has a very long lifetime of a few hundred nanoseconds in a scattering lipid environment. And if you look on this, we open the light, you see nothing. And as we shift the gate, you see the tubing this showing up out of the scattered medium. So that's a power of gating, but it's a passive technology at this case. So for a long time, we were asking ourselves how we can increase the signal of the probe, and mostly how we can eliminate the background. And now we realize a couple of things. If you do the excitation and you have short and long component, what you measure something which is a sum of all of them. And at this point, that would be intensity decay. And there's nothing you can do. If you do CW excitation, it's mean continuous wavelengths, the ratio between the parameters is constant. So it doesn't matter how you manipulate this, you always measure the same ratio between your signal and your probe. You only can do the gating, which is passive, and that's what gives you advantage. And then not long ago, we realized we can do a very simple trick, because at this point, we ask ourselves, what will happen when after some time, when this short component decays, we put another pulse? Of course, it will excite the short-lived molecules and it will excite the long-lived molecules. One thing which you have to remember, these molecules, long-lived molecules up to this time, they don't have memory how they were excited. So wherever I start measuring the lifetime, it always would be the same. So they don't know how they were excited, they were just randomly emitting. So eventually, when you look on this, if I put a set of pulses properly separated, I am increasing the population of long-lived component. And depending on the lifetime, the increase is proportional. And in case of short lifetime, you can see that the same pulses will always produce the same population of a short lifetime component. And you can see, depending on the lifetime, we can pump what we call this quite significantly. And now it comes to a very simple thing, how we want to measure. And its important thing is to realize that time-correlated single photon counting, and I didn't have time to talk about this, but I hope you have some idea how it works. You can trigger your measurements as you like. You can shift the trigger as you want. So we always trigger with the time 0 here, what we call time 0. And in case of single pulse, that's what we measure. But in case of multiple pulses, in this case 4 pulses, we should first kind of priming pulses, and on the end, on the last pulse, we start the measurement. And at this point, you realize that in every time, we're starting with the same fraction of a short component, but the long component fraction increases quite a lot. And eventually the cycle repeats. So you have essentially, instead of single pulse, you have multi-pulse excitation. And now we try a lot of things because we ask ourselves, this will perturb the system, how much will change, what can happen. So this is rhodamine measurements with 1 pulse, multiple pulses, always the same lifetime. This is ruthenium, much longer lifetime, doesn't matter how we do it, it's always the same lifetime. The only difference is in case of ruthenium, here is logarithmic scale, so it doesn't look so dramatic, but in linear scale, you can see with 10 pulses, you can increase almost 10 times the initial intensity of your long-lived probe for the measurement. We did a very simple measurements with the beads, published long time, almost 10 years ago. And we can see that depending on the number of pulses, 1 pulse, 2 pulses, I think 4 pulses, 8 pulses, 20 pulses, we can see brighter and brighter spot. We've seen some background rays, which was surprise at this time for us, but now we know that the beads were leaking the dye. When we did the tissue experiment, this is essentially ocular tissue, where we did a nice imaging and we look for the tissue. With this pumping, we were able to pump this quite significantly. And it's quite nice and quite easy to do it. So it's easy in the case of really long lifetime, like ruthenium, which is a few hundred nanoseconds. So that's kind of an obvious to do. Can we do something with the shorter lifetime? Especially when we realize that a lot of markers like ethidium bromide for DNA changes the lifetime from 1.5 nanoseconds to 20 nanoseconds. INS from much below 1 nanoseconds to 20 nanoseconds, or like I showed you already, acrid in orange, from 1.7 to 20 nanoseconds. everything is simple, but we have to realize that the second pulse must come pretty early. So you have to have a high rep rate of the pulses. In case of ruthenium, we can use 80 megahertz, and the separation between the pulses is 12.5 nanoseconds. But this is for a lifetime of 15 nanoseconds, much too long. You will not pump too much. So we worked for a while how to generate the bars of pulses, and it's not easy to buy anything which will generate more than 80 megahertz. And we find out a very simple way, assembling on the table where we can separate the pulses, put through the different trajectory, and 1 foot is a 1 nanosecond. So it's an advantage for us. If we want to have 3-4 nanoseconds, we just change the delay lines on each trajectory by 3 or 4 foot, and we have essentially delay of the pulses. And this is the example of a long-lived dye, which is 20 nanoseconds lifetime only, with 5 pulses, and you can see how the intensity increases quite significantly. And when we start measurements here, we have a huge advantage of the intensity of the probe. Later on, we came up with a very interesting design of the fiber, where eventually fibers splits to essentially 4 different lens fibers, differencing by roughly 2 feet. And at this point, the delay is 4.5 nanoseconds between each pulses. So we put a single pulse here, and eventually this pulse comes as a 4 separate pulses separated by around 4.5 nanoseconds. Immediately, you can see the application to this example where the lifetime is practically 15 nanoseconds. So if we put, we can do the enhancement with this part, and we can do this quite easily. One thing which I want to show you, it would be practically application to detecting the DNA, because that was one thing which was kind of interesting for us. And in collaboration here with Forensic Institute, we realized it's a very big need to detect the DNAs on surface or any places, because that becomes a marker which we can amplify with PCR, and again detect this with fluorescence. But after amplification, you have huge amount of DNA. But in practice, if you want to collect, DNA is colorless. You don't see the stain with DNA, and you know it's typically very limited amount. What kind of part we can do? And one of the example I want to show you. We want to do this without any processing and just detect the DNA. And this is one example we did, one of the very first part, where we use ethidium bromide, and we use this aspect that yes, the lifetime is 20 nanoseconds. We can pump it, and we can detect this with pretty significant part. To make it a little more sequencing, we did a nice application with detecting just the spectrum, now without the pulses, but utilizing a change in the lifetime. And this is, if you want to detect a very low concentration of DNA, you have to remember your dye, which you use for the detection, constitute your background. So you have to have a dye concentration also very low. And in this case, you can see we have so low concentrations that we can detect the fluorescence of our dye when it's free or bind to DNA. But in the same case, your system is worked so hard that you also have the Raman scattering. And this was excitation 475. This was a Raman scattering, but you can always do the spectral decomposition, and you can really detect spectrally quite easily. And this is the example of the system where we were doing this detection. It's just an excitation line, a tip of a swabbing, of a swab, and you can detect and you can characterize this. And the idea was try to detect the DNA. And this is the example where we have 3 swabs illuminated with light. If you make a picture when they are clean, that's what you see. And then you deposit a small amount of DNA on one of the tip and do exactly the same detection. And you can say, yeah, the DNA is in these places. And the value is huge, because if you process entire swab, your DNA is, your amount of DNA is constant, but all the perturbation from the swab material and everything else becomes a problem. So if you can extract the DNA only from the places where is the DNA, your amplification process becomes much easier and much more clear. With the pulses, we are getting this advantage that if we put the pulses separated by 4 nanoseconds, this is with our fiber, you can find out eventually that you are pumping the long-lived component. So you're only pumping this component which binds to DNA. This gives you a huge enhancement in the detection. And at this point, you can see you can detect quite significantly. And this is a practical application of this kind of pulses. This is how this pulses comes from the fiber. You can see they are not perfectly equal. You cannot make a perfect fiber at this point. But eventually we can do 1 and 4 pulses. And at this point, when you look on this, this is an imaging camera, which the camera which I was showing you before. It's essentially can take 10 million frames per second, and we can shift the opening time as we like with the precision of few hundred picoseconds. But the opening time of the gate is kind of a slope, so eventually your resolution is within 5 to 10 nanoseconds if you're shifting the gate. And eventually you can see this is a cap from the 1 centimeter cuvette where we drill tiny holes of 3 holes here. We put essentially ethidium bromide to all 3, let's say water, ethidium bromide to this 2, I think. And at this point, we were adding a different amount of DNA and doing the imaging where eventually we were putting a single pulse or multiple pulses and looking how well we can detect. So if we put DNA to water, of course, it will not be changing much. If we put if we put DNA to ethidium bromide, it will change significantly. And that's what we can do this with the detection. This is the image. At this time, we were able to go down to around 400 picograms of DNA. To give you an idea how much it is, it's essentially a DNA content of a single cell is in the order of 6 to 10 picograms. Now, to give you an idea how many cells we can expect, a human loses roughly 10 to the 11 cells per day. It's an incredible number. And within a minute, you can lose up to 30,000 cells. It depends on the individual. Some, what they call the shedders, lose more, and less shedding people lose less. But my talk, which is almost 1.5 hours, you lost probably close to 1 million cells, which at this point can be easily detected. And eventually one thing which came up very recently, which we realized that it could be eventually done with a very high precision. And this is a kind of simulation where I put a short-lived background, which is this black line, and red, that's a simulated spectrum, so don't worry about this, which has a longer lifetime of 20 nanoseconds. If I mix 10% of this one to this one, that's how the spectrum will look like. That's how the lifetime will look like. In principle, when I do the spectral decomposition, I could do it, but it would be very difficult to detect. And now I want to apply the conditions where I have a single pulse excitation and 4 pulses excitation, and on top of this, I can use the time-gated detection. So if I do a single pulse, when I measure in regular ways, that would be the spectrum I will measure. If I put 4 pulses, I amplified this component, but I don't amplify this one, and now I will measure much better. And you can see the scale is different, so that's what is important. Now the question comes: what makes this difference? The difference between this and difference between this is essentially this long component here. So what will happen if I take this spectrum, the dashed blue, and this spectrum, and from this one I subtract this one? And a simple subtraction shows you that this is what I had with the single pulse. This is what is subtraction, 4 pulses minus 1 pulse. I have clear only the spectrum of my long-lived component. We did this practical application with different separation of the pulses because the lifetimes, it depends. And for short component, we can do some amplification, which will show up like this one. To give you a practical application for long lifetime, we took 3 nanoseconds rhodamine and ruthenium, 500 nanoseconds. We mix them, and we mix them in different proportion. When I talk 50-50, you can see this kind of depending on number of parameters. When I change the proportion, I detect a little bit, and for this one, it's practically nothing. And if I measure the same spectra, they are kind of time result spectra, measure it with 1 or 4 pulses, you will find out even for this mixture 19 to 2 with 8 pulses, I clearly see the ruthenium. So this gives us a huge power at this point. So what's happening if I take this kind of a subtraction? Let me check if I have. If I take now this pumping and do the subtraction, I can find out that I can find out the lifetime of my long-lived component of ethidium bromide. And at this point, it's a kind of a nice experiment which shows us that by subtracting the spectra, we can eventually detect only long lifetime component. How it works in the regime of imaging. And this is a kind of a mode of experiments. You have 2 films, ruthenium and rhodamine, which we excite this on this little dish. And at this point, if you look on them, that's how you can see it with different number of pulses. And this is 8 pulses. After subtractions, it becomes only ruthenium. And now we put 2 ruthenium films on a huge background of rhodamine. If you excite them, normally you see these traces of the film, but this is dominated by the fluorescence of the rhodamine. With 8 pulses, you can see that this film becomes visible, and when we subtract what you left, you only left with the films you wanted to image. And then we went a little further. We said, let's do the perturbation in sense of scattering. So we put the same background with the same films into the dish where we put the intralipid solution of different volume. And you can see how it's happened. If you do single pulse, you see the background and everything. As you go do the subtraction, you can see better and better. And eventually here, for much higher concentration of intralipid, visually you can see trace of this what you put it. If you illuminate from the surface, it disappears because it's scattering on the surface so high. And now if you increase the number of pulses, you can see it shows up pretty nice. It's kind of diffused image, but that's what you expect because your fluorescence photons are also scattered a lot. So that would be a very important thing to remember. And at this point, when you look, we did this experiment with chicken drum, where you eventually take the ruthenium and rhodamine film, you put this and you cover with the skin. And I want to mention it's 375 excitation, which is practically barely penetrating the skin when you do the excitation, but still do and produce a lot of autofluorescence of the tissue. So this was experiment against us, where we had maximum scattering and maximum autofluorescence. And when you cover, that's what you see. Barely you see the both films. And now 8 pulses, you still see much better the rhodamine film. And if you do the subtraction, all the scattering goes away. Let's say scattering part, and you see this film pretty nicely. Now, the spectral subtraction gives us a huge advantage because right now we can do a number of things. We can perturb the spectrum by number of pulses, or we can perturb by any other process. And one of the processes which can change the emission is energy transfer. And in this case, in this experiment, we use the ethidium bromide solution with DNA, to which we added a little bit of DAPI. And DAPI is an intercalator dye which binds pretty strongly to DNA, stronger than ethidium bromide. So if we add the DAPI, and we are within the excitation of range where we can excite the DAPI, adding the DAPI gives us the same possibility. DAPI intercalates to the DNA. What happens as it intercalates, it comes closer to the ethidium bromide, and now we have a linear system which can transfer the excitation energy from this one to this one. And measurements at this point, it was very simple. We took the DNA with ethidium bromide, and eventually to one of these loops, this is water, we added a little bit of DAPI, and this becomes much more brighter. We change nothing, just a little bit of DAPI, and at this point, when we do this subtraction, we can easily detect the image. Easily, you can see this barely image, but this is 100 picograms of DNA. So now we are within 10, 20 cells. That's what we can detect, and we can visualize the DNA on this part. And now we added also pulses, and we use a little different part, which is a yo-yo. This is something which is now being on the process of submission. And we do the pulse subtraction and doing this spectroscopically. So working with the spectra where we have pretty good control, you can see we are within 10 picograms of DNA. So the amount of DNA from single cell is sufficient. What is really important, we can do a single DNA strand imaging right now, but these are single molecule regime. Here we are doing the detection without any purification or anything. It's on anything which has just DNA. And here is the spectral analysis and imaging. And at this point, we can see nicely spectrally 10 picograms, but on the imaging, we are within 50 picograms. So we are within 5 to 10 cells where we have left. And if you leave a fingerprint on something, you are leaving quite a few number of cells. We are talking about 100 cells easily. So at this point, we can just using this technology, and it's a very simple approach. You put ethidium bromide, which does not show anything because it's not a very good marker, but you make an image, you add a little bit of yo-yo, and then you take this image and the previous image, you subtract, and what you left with is essentially where you had a yo-yo. You gain two things. You increase your sensitivity, And most importantly, you increase the specificity for DNA, because in this case, both dyes have to be mixed, have to be within the DNA helix, because otherwise there will not be any kind of threat. And just to close my talk, it's a little of applications which we are developing right now, where we want to do the pumping and image subtraction in surface plasmon couplet emission, where we eventually use the near field interaction for the excitation of the molecules, and eventually trying to do all these aspects of detection on the surface. This is for immunoassay, where we can eventually incredibly limit the volume, excitation volume over the surface, which eventually comes to much below 100 nanometers, and all the backgrounds is decreased. And now when we do the pulsing and subtraction, we can really see very, 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 very small concentration of any kind of markers. And just one thing which this technology of pumping becomes extremely useful is detecting of phosphorescence. And I just want to mention one recent thing which happens, because if we go with fluorescence, you excite the phosphorescence, you excite the single system, singlet state, intercross system comes to the triplet state, we have phosphorescence. It's very nice process, long lifetime, especially for the proteins, where we can excite phosphorescence, but in sense of polarization and applications is quite useless, because fluorescence always dominates phosphorescence, because at the beginning it's million-folds higher than phosphorescence, and intersystem crossing eventually changes the polarization. The most recent things which we did, we asked ourselves, can we do direct excitation of singlets, of triplet state? We did a lot of trials. We always hope with Ignacy, with my brother, we were doing this for 20 years. And eventually, recently in PVA film, we were able to do this for indole derivatives, for tryptophan, and most recent things which is going right now is benzoquinolone, where we are showing that this excitation is possible. What this gives us? It's a huge advantage, because this excitation and phosphorescence process, it becomes like fluorescence, high anisotropy, 0.4. That's what we can measure. So we may have excitation of the tryptophan, which gives us a very long lifetime, so we can study really big molecular complexes, and we have eventually very high polarization. And another aspect, we don't excite singlet state. So we have a tryptophan, which typically when you wanted to have phosphorescence, you excited at 290, and your phosphorescence was around 450. Now we come with the excitation 400 or 405, because it's a laser, or 375. And we don't excite singlet state, we excite only triplet state, no perturbation from the singlet state, because thermal activation to this way in room temperature is not efficient, and we can really observe pretty nice phosphorescence. So I think this could be something nice, really big for the future of application of phosphorescence and to fluorescence field of biomedical field. So thank you very much, and I will be happy to answer any questions.
Poznan University of Technology.